Malaria is a potentially life-threatening acute febrile illness caused by a protozoan parasite called Plasmodium. 5 species of Plasmodium are known to cause human malaria, including Plasmodium falciparum, Plasmodium vivax, Plasmodium ovale, Plasmodium malaria-an and Plasmodium knowlesi. Plasmodium falciparum and vivax are the most common species worldwide, but Plasmodium falciparum is the most prevalent species in sub-Saharan Africa. Malaria is endemic in most parts of sub-Saharan Africa and in some parts of South America and Asia. However, some countries in this region are reporting 0 cases of malaria, and a few others have recently been certified malaria-free. No indigenous cases of malaria have been reported in Europe and America, and parts of Asia. Human malaria is transmitted by blood-sucking Plasmodia-infected female Anopheles mosquito. There are over 480 species of the Anopheles mosquito, but only about 80 are known to transmit malaria in nature. In Africa, malaria is transmitted by 2 major species of mosquitoes, namely Anopheles gambiae complex and the Anopheles funestus group. Adult Anopheles have slender bodies with 3 sections: head, thorax, and abdomen. The head contains the eyes, a pair of antennae for detecting odors, a proboscis used for feeding, and 2 sensory palps. The thorax has a single pair of wings and 3 pairs of legs. The abdomen is a segmented body part specialized for food digestion and egg development. Anopheles mosquitoes can be distinguished from other mosquitoes by the palps, which are as long as the proboscis, by the presence of discrete blocks of black and white scales on the wings, and by their typical resting position. Anopheles adults rest at an angle of about 45 degrees to the surface they are standing on, with their abdomen sticking up in the air, while adult Aedes and Culex rest with the body parallel to the surface. Mosquitoes breed in warmer and more humid climates but require water to breed successfully. Any standing or slow-running water is a potential site for mosquitoes to breed. This will include water left in flower pot trays, water around sewage areas and uncovered trash cans, old tires, and air conditioner drip trays, as well as puddles of rainwater, blocked drains, slow-running streams, untreated pools, broken pipes, and uncovered water tanks and buckets. Anopheles mosquitoes go through 4 stages in their breeding life cycle: egg, larva, pupa, and adult. The first 3 stages are aquatic and last 7 to 14 days depending on the species and the ambient temperature. Adult females lay 50 to 200 eggs during each cycle. Eggs hatch into larvae which mature into pupae and then to adult mosquitoes. The maturation from egg to adult may take as little as 7 days but usually take 10 to 14 days in tropical conditions. After its body parts have hardened, the adult mosquito emerges onto the water's surface and flies away, ready to begin its life cycle. Adult mosquitoes usually mate within a few days after emerging from the pupal stage. In some species, the males form large swarms, usually around dusk, and the females fly into the swarms to mate. After mating, the male and female mosquitoes feed on nectar and other sources of sugar for energy, while the female mosquitoes fly in search of a blood meal from humans or animals to develop their eggs. Anopheles mosquitoes may be active at dusk or dawn or nocturnal. Some Anopheles mosquitoes feed indoors while others feed outdoors. They are attracted by body odors, carbon dioxide and heat emitted from a potential human or animal host. When they find a host, mosquitoes use their proboscis to draw blood. After feeding, a blood-fed mosquito looks for a safe resting place to digest the blood meal and produce eggs. The anophelines that enter houses to feed often rest indoors for a few hours after feeding, but some prefer to rest outdoors on vegetation or at other natural sites. Mosquitoes do not usually bite while eggs are developing. Once the eggs are fully developed, the gravid mosquitoes leave their resting sites and try to find a suitable breeding habitat. After laying their eggs, they fly in search of another blood meal to develop another set of eggs. The cycle repeats itself until the female dies. Females can survive up to a month, or longer in captivity, but most do not live longer than 1 to 2 weeks in nature. The life cycle of the malaria parasite takes place in the gut of the female Anopheles mosquito and in the human body. The mosquitoes are the definitive host, while humans and other animals are the secondary or intermediate host of the parasite. After identifying a suitable human host, a Plasmodia-infected female Anopheles mosquito draws blood using its proboscis and at the same time inoculates sporozoites from its salivary gland into the human body. The sporozoites are carried by the peripheral blood circulation to infect the liver cells. Within the liver, sporozoites grow and mature into schizons. The liver schizons produce thousands of myrizoites and then ruptures, releasing myrizoites into the blood circulation. In Plasmodium vivax and ovale infections, liver schizons may transform into a dormant stage called hypnozoites. Hypnozoites do not cause symptoms, and if untreated, they can persist in the liver for weeks to years and then cause relapses of malaria by invading the bloodstream. The initial replication of the malaria parasite in the liver cells is called the exoerythrocytic schizogony. After liver replication, the parasites undergo asexual multiplication in the erythrocytes or red blood cells. Myrizoites infect red blood cells and transform into ring forms, and then ring forms transform into trophozoites. Trophozoites multiply and mature into schizons. This blood stage schizons then rupture, releasing thousands of myrizoites, which can infect other red blood cells and repeat the cycle. This stage of replication of plasmodia in the red blood cell is called erythrocytic schizogony. The erythrocytic cycle is responsible for the clinical manifestations of malaria. Sexual life cycle of the malaria parasite begins when some parasites differentiate into sexual erythrocytic stages, also called gametocytes. The gametocytes, male, microgametocytes, and female, macrogametocytes, are ingested by an Anopheles mosquito during a blood meal. The parasite's multiplication in the mosquito is known as the sporogonic cycle or spogony. While in the mosquito's stomach, the microgametes penetrate the macrogametes, generating zygotes. The zygotes in turn become motile and elongated, eukaryotes, which invade the midgut wall of the mosquito, where they develop into oocysts. The oocysts grow, rupture, and release sporozoites, which make their way to the mosquito's salivary glands. Inoculation of the sporozoites into a new human host perpetuates the malaria life cycle. After an incubation period of 7 to 23 days, humans infected with plasmodia may present with asymptomatic malaria, uncomplicated malaria, features of malaria in pregnancy, or severe malaria. Asymptomatic malaria is defined as malarial parasitemia of any density, in the absence of fever or other acute symptoms, in individuals who have not received recent antimalarial treatment. Asymptomatic malaria commonly occurs in partially immune adults in malaria endemic countries. Uncomplicated malaria is symptomatic malaria parasitemia without signs of severity or evidence of vital organ dysfunction. The common features of uncomplicated malaria are fever, headaches, fatigue, malaise, nausea and vomiting, and other nonspecific symptoms. Malaria in pregnancy may present with symptoms of uncomplicated malaria and may lead to stillbirth, preterm birth, anemia, miscarriage, and low birth weight. Uncomplicated malaria could progress to severe malaria, especially in children and non-immune adults. Severe malaria is defined as symptomatic malaria with signs of severity or evidence of vital organ dysfunction. Some clinical manifestations of severe malaria include cerebral malaria, defined as coma for at least 30 minutes with or without convulsions, impaired consciousness, renal failure, prostration, shock, severe anemia, multiple convulsions without coma, and pulmonary edema, among other features. Delay in treatment of severe malaria could lead to death. The prevention and control of malaria requires multiple integrated strategies, including avoiding mosquito bites, reducing or eliminating mosquito breeding sites around homes, taking malaria chemoprophylaxis when needed, seeking medical attention early if symptoms develop, and using a malaria vaccine where available. Avoiding mosquito bites could be achieved by wearing thick, bright-colored long-sleeved clothing and trousers, especially when outdoors at night. Indoor residual spraying, use of insect repellents and insecticides, will kill adult mosquitoes that rest on walls within homes. Insect repellents may also be used on skin to protect against mosquito bites. Using a mosquito net, preferably a long-lasting insecticide-treated bed net, will provide a protective barrier against mosquitoes, and may repel and kill mosquitoes if insecticide-treated. Screening windows and doors will also prevent mosquitoes from entering homes. Where available, air conditioners can prevent mosquito bites by creating a cold, unfavorable environment for mosquitoes to fly in by dispersing heat and odors required for mosquitoes to locate a potential human host. To reduce mosquito breeding sites, eliminate all standing or open water around homes. Make sure all tanks, water deposits and containers are covered and sealed to keep out mosquitoes. Get rid of old tires around homes. Clean drains and gutters and ensure they are flowing freely. Pour out water from flower pots and planters and replace with damp sand. Safely dispose of any unused containers and objects that can accumulate water and serve as breeding sites. Provide a covering over dump sites to protect waste from rain. Keep swimming pools adequately treated with recommended products. It is important to seek early medical diagnosis and treatment if symptoms of malaria develop. Early treatment will reduce the spread of the infection to others and avert progress to severe malaria, especially among pregnant women and children who are at a higher risk of symptomatic malaria. Malaria vaccines are designed to target various aspects of the parasite life cycle in humans. There are 3 types of malaria vaccines in development, including pre-erythrocytic vaccines, blood stage vaccines, and transmission-blocking vaccines. The pre-erythrocytic vaccines produce antibodies that kill infected liver cells or interfere with the malaria parasite during liver cell proliferation. Asexual blood stage vaccines are primarily aimed at reducing parasite load or eliminating circulating parasites. Transmission-blocking vaccines are designed to control the transmission of malaria parasites from human hosts to the mosquito vectors. Although several candidate malaria vaccines are undergoing trials, a pre-erythrocytic vaccine named RTS-SAS01, also called Mosquitoes, is the only malaria vaccine currently licensed for use by the World Health Organization. The WHO has recommended widespread use of this malaria vaccine among children in sub-Saharan Africa and in other regions with moderate to high Plasmodium falciparum malaria transmission. The recommendation is based on results from an ongoing pilot program in Ghana, Kenya and Malawi, which has shown that the vaccine reduces the incidence of severe malaria in children by as much as 30%. The vision to have a world free of the burden of malaria in 2030 can only be achieved through deliberate efforts and investments that will lead to elimination of malaria and prevent re-establishment of the infection in every country of the world.